Collecting arthropods involves careful preparation to ensure effective sampling. Today, we'll discuss the materials used and how to prepare and set baits for attracting arthropods. For this collection method, you'll need an insect net, a killing jar, and bond paper for the bait. The insect net is essential for catching flying arthropods, while the killing jar, usually containing a chemical like ethyl acetate, is used to quickly and humanely euthanize the collected specimens. Lastly, bond paper will serve as a surface to hold the bait mixture. Now, let's move to the bait. A mixture of peanut butter, tuna, and seeds works effectively to attract a variety of arthropods. Peanut butter offers a high-energy food source, tuna provides strong scents and protein, and seeds attract herbivores. Spread this mixture evenly onto a piece of bond paper. Place the baited bond paper in areas where arthropods are likely to gather, such as near plants or damp soil. The aroma of the mixture will attract various arthropod species. Check your baits regularly, using the insect net to catch any flying arthropods, and transfer them to the killing jar for preservation. By using an insect net, a killing jar, and a simple bait set up with bond paper, you can effectively attract and collect a diverse range of arthropods, ensuring successful sampling for your study.
For our arthropod collection, we place 8 sets of bait traps across various indoor and outdoor locations around the FEU campus, using the pre-made peanut butter and tuna baits to attract specimens. Out of the 8 collection sites, 5 traps successfully attracted ants, mostly red, of varying sizes. The outdoor sites showed a much higher level of activity compared to the indoors. To collect and preserve the specimens, we sprayed them with alcohol before individually storing them inside Ziploc bags. In total, the mean of the roughly counted ants was 32, with one site alone yielding an impressive count of 183 ants. As for our sweep net collection, it was less productive, with only one moth and a caterpillar captured. Both were placed into a kill jar for preservation. This collection will now undergo further analysis to identify the species and better understand their distribution on the campus.
To identify the ant specimen, first we have the material, the ant specimen, which is collected in a suitable environment, and the second is the microscope. This is the dissecting microscope or compound microscope for the detailed examination, and petri dish for observation and manipulation of the specimen, forceps for handling the specimen without damage, and labeling materials, small labels and pen for the documenting specimen details, and the camera for taking images for their references. For the procedure after the collection and preservation, the observation used a microscope to observe the key features such as the antennae, mandible, and the body segmentation. And after that, the identification, we use the dichotomy scale to narrow down the species based on the observed characteristic. And after that, with documents, we record the details about each specimen, including the collection's location, date, and the environmental conditions.
We identify the specimen based on the book as a synoptic review of the art general of the Purines by David General and Gary Albert. These books help us to identify the subfamily and genera based on the morphological characteristics. Under the microscope, at the profile view, we look at the presence of host features and the states that we not see in this specimen. Also, the acetochord is absent, which is the apex cluster with a transverse slit-like orifice and the antennal socket, always abutting the posterior margin of the flying face. So we assume that this ant belongs out to the Continae subfamily. So proceed to the genus. We determine it as Typhinoma because of the duster with the 4 dorsal segment, and the 5th segment is reflex.
You'll need a few essential materials to pin an ants. Let's start first with the entomological pins, which you can buy on Lazada, and a pinning block to standardize the height of your specimen. Next is a 1/4-inch styrofoam sheets that provides a stable base. Also, you'll need paper tips that is created using an entomological puncher, which are useful for mounting ants that are too small to pin. Next is security ants with the water-based glue or Elmer's glue. Also, you'll need an iguchi ball. It is a weighted ping-pong ball filled with a mixture of cement and clay for delicate handling. Also, don't forget to provide a data label for determining the taxonomic details. Finally, ensure your ants is mounted carefully for study and preservation.
For procedure and sample of clean specimen, first the sample is softened with 70 to 75% alcohol placed in the petri dish. Next, we carefully studded the paper tips using the entomological pins with the help of the pinning block. After that, we placed the stage pin in the iguchi ball for further pinning of the ant sample. And now we placed the iguchi ball with the stage in the microscope and added the water-based glue with enough amount in the tip of the paper stage. After that, we now position the ant on the stage under a microscope using the probe to avoid damage and for easy proper pinning of the ant. Furthermore, adjusting the legs and antenna with the probes for clear visibility was also done carefully. And once the ant is positioned properly and all the parts are visible, we now label the stage with the species name, collection date, location, and the collector's name.
Dissecting a mosquito proboscis requires a careful and precise approach using specialized tools like fine forceps and entomological probe under a dissecting microscope. We will begin by preparing a clean workspace and placing the freshly catched mosquito onto a petri dish with a small amount of water as a solution to prevent desiccation. Under the microscope, we will secure the mosquito in position and gently hold it with the probe. Using the probe, gently pull at the base at the outer sheet of the proboscis to expose the other mouth parts. Carefully, we will separate the labium, which is the outer sheet, and from the fascicle, which contains the feeding stylets, ensuring not to damage the delicate structures. We first examine the head and thorax of the two mosquito specimen. The first specimen from the genus Ulysses shows a plumose antennae indicating that its gender is male. The halter is also present, which functions as a rapid feedback to the wing steering muscles responsible for stabilizing the mosquito's flight. The second specimen is also from the genus Ulysses and the sex is female. In Ulysses females, the palps are much shorter than the proboscis, while in males the palps are usually as long as or longer than the proboscis. The mosquito proboscis is a specialized feeding organ composed of several needle-like parts designed for piercing skin and sucking blood. It includes an outer sheet called the labium, which bends back to expose the inner structures during feeding. Inside, the labium forms a food canal for drawing in blood, and the labella at the tip help the mosquito locate blood vessels. While the mandibles and maxillae are serrated tools used to cut through skin. However, we did not have a clear shot of these serrated tools since the specimen's proboscis is too brittle to cut. Also, we did not saw the hypopharynx that is responsible for releasing saliva containing anticoagulants to prevent blood clotting. All in all, some mouth parts of the mosquito has been seen and been recorded for the said activity.